Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I, of course, am Mr. Ulrich. In this notescast, we're going to build on what we've been talking about as far as the structure of DNA and the function of DNA, and actually get into how DNA acts as the instructions for protein construction. And if we're going to talk about the construction of proteins, then we need to talk about what proteins are and what they're made of, right? Well, they're huge molecules, generally speaking. They are polymers in that they are chains of repeating units. In this case, the repeating units are amino acids. We're going to get to the different types of amino acids that are out there later on in the video here. They are ubiquitous. They're found throughout all of cells of all organisms because they're involved in every metabolic process, essentially. We can loosely group them together into two categories, being structural proteins, things like keratin that makes up hair, and collagen that holds our cells together, as well as actin and myosin that makes up our muscles. Of course, keratin also makes up things like duck bills and nails and stuff. And then we also have regulatory proteins. These are proteins that help maintain homeostasis, like hormones for communicating from one part of the body to another, as well as neurotransmitters, enzymes that are going to be catalyzing all of the chemical reactions that take place within cells and outside of cells. And oh my, that's a lot of chemical reactions. And transport proteins, things like hemoglobin. Just like when I introduced the structure of DNA, we started out with what a nucleotide is. When I introduced the concept or the structure of proteins, we need to talk about amino acids, the monomer of the polymer. Amino acids are a group of molecules that have particular functional groups in common. A functional group is a group of atoms arranged that we see really commonly in lots of different molecules, and they impart certain characteristics. Over here on one side of an amino acid, we have an amine group. It's an NH2 group. It sounds like ammonia because ammonia is NH3, and this is NH2. Anyway, there's a central carbon that amine is attached to. That central carbon is also attached to a carboxyl group. And a carboxyl group is acidic. It makes a compound acidic. And so when you put these together, you get an amino acid. Now that's what all amino acids have in common. The amine, the central carbon, and the carboxyl group. What gives each amino acid its own kind of personality is this other functional group, or R group, that's attached to the central carbon. There are, in most living things, there are about 20 different amino acids, and that means that there are 20 different functional groups. It's the side chain then that gives each particular amino acid its particular characteristics. And we can group side chains kind of together by their similar characteristics. This table shows these side chains over here are all positively charged. These are all negatively charged. These amino acids over here don't carry a charge on them, so they're not positively, they're neutral. However, all of these are going to mix pretty well with water. These amino acids down here are what we call nonpolar. They are hydrophobic. That means that they don't mix well with water. Just like when we start putting nucleotides together, we form a polynucleotide. We start putting amino acids together, we start forming a polymer. It's a polypeptide. The reason why it's called a polypeptide is once we glue one amino acid to a neighboring amino acid, it's this process called dehydration synthesis, which you'll get to in a regular bio class. A special type of bond is formed called a peptide bond. So as I start bonding amino acids together, I'm going to get a chain of amino acids and a chain of peptide bonds in between them. As I do this, I'm going to start getting, essentially, as in this diagram here, I'm going to start getting a chain of side chains, right? And each different side chain here is going to have a particular characteristic. And like I said, some of these like these guys here, are going to mix very well with water, and this one too. Some of these are not going to mix very well with water. These guys here aren't going to mix well with water. So when I start putting polypeptides into water solutions, they're going to start to kind of fold up and bend so that the parts, the oilier parts, the nonpolar parts as we call them, like these guys right here, are going to want to come together in the middle and stay away from the water. And then the charged and polar parts, here's another polar one here, are going to want to go into the water. And what that's going to do is it's going to cause that straight chain to kind of curl and put the nonpolar ones inside and put the polar ones on the outside. And we start to get, instead of this long, nice chain of amino acids, we really start to get bends and loops. And plus, if you notice, here is, let me change color a little bit. Here's purple. Here is a positive charge, and here is a negative charge. And we all know that opposites attract. And so, once again, when we put this in water, not only are these going to orient themselves out towards the water, and these guys here inside towards themselves, right, to get next to the other hydrophobics, there's going to be an attraction formed between these two, an electrostatic interaction that's going to bend that chain of amino acids into weird contortions and shapes. And so we end up with helixes like we see in this region here, and other kind of, you don't get a chain necessarily, you get kind of a blob in a lot of cases. This brings us to the central dogma of modern genetics. I like to back it out and even call it the central dogma of modern biology myself, because it's that important. If you want to know why we call it the dogma, look up dogma. We can discuss that in class. It's a pretty interesting discussion to have. But we need to stay focused here on what's going on. So it goes like this. We start out with the base sequence of DNA. Right, all those A's, T's, G's, and C's. And most of you came to me understanding that DNA makes you who you are, right? DNA makes me who I am. And that's a fine understanding for up to grade school, middle school. Now we're highfalutin high school students. We need to get a better understanding of what goes on in between the base sequence of DNA, between DNA and who you are and your traits. And that's kind of where we're going. So the sequence of A's, T's, G's, and C's directly determines the sequence of amino acids, all those different 20, 22 different amino acids in a particular protein. It's the sequence of amino acids that's going to determine, by all of those interactions with the water and the bonding between themselves, that's going to bend a particular polypeptide or chain of amino acids into a particular shape. And just like keys have a particular shape, the shape of the protein is going to determine the individual function of those different proteins. And your traits are the result of the functions or lack of function of the particular proteins that you have. Now, you're going to talk more about proteins in a regular bio class in biochemistry, and you're going to talk more about the inheritability and how traits are passed on from one generation to another generation. In genetics, in fact, we'll go over this molecular genetic stuff again because it's that important and it's that confusing. But for right now, we are going to focus on how we get from the base sequence of DNA to the amino acid sequence of the polypeptide of the protein. The problem is in a eukaryote, which are cells that have nuclei, among other things, all the DNA hangs out in a nucleus. The places where the sequence of bases is converted or translated into a sequence of amino acids is out in the dangerous cytoplasm at structures called ribosomes. All right, so the problem is if the DNA doesn't leave the nucleus, how does the code get used at the ribosome? Well, we have to make a copy. Right? And so the DNA, the code of DNA, stays safe in the nucleus, and we make kind of a temporary copy of the gene into a different compound, a different nucleic acid called RNA. In particular, this is called mRNA, all right, or messenger RNA. It's what sends the message of the base sequence to the ribosome. It's temporary, right? Uses the same code of nitrogenous bases, right? Or it uses nitrogenous bases to encode the info. There is a difference where instead of A's, T's, G's, and C's, it uses uracil instead of thymine. So it's almost the same as DNA, but not quite. All right, thymine is a perfect stand-in, uracil is a perfect stand-in for thymine. Right. I refer to it as the PDF of protein synthesis. It is the portable document of a gene. It's not meant to last very long. It's kind of temporary. Once it's built, it goes out there, and then eventually it gets chewed up by enzymes in the cytoplasm. There are other types of RNA, and we'll get to those in a bit. So this is actually the first step of making a protein. It is making that strand of messenger RNA with the same base sequence as the strand of DNA. We call this transcription. Transcription just means to write something kind of over there. So you're just really copying something over. And just like in music, when you transcribe, you're really just changing keys, right? It's still notes. It's still, you know, A's and B's and C's and D's and F sharps and B flats and stuff. All right, but that's just kind of moved over a little bit, right? In this case, it's still A's, well, U's, G's, and C's. It's still it's still nitrogenous bases, but slightly different. This is going to take place in the nucleus because that's where the DNA is going to stay. And if it looks like replication, that's because the process is very similar to replication. The big difference is that only one new strand is being formed. If it was replication, I'd get another strand forming over here, but I'm not forming another strand over here. I'm just forming this one new strand on this one side. All right, so one side acts as the template, it's called. So that's the template strand. As that messenger RNA is synthesized, it can kind of make its way out of the nucleus through a little structure called a nuclear pore complex, make its way out of the nucleus into the dangerous cytoplasm. Eventually, it will wander around, bump around, and complex with ribosomal subunits, right, and make a ribosome. Please, this ribosome is grossly enlarged for your viewing pleasure. They are usually not this size. They are two subunits. One is bigger than the other, so it looks a little bit like a lopsided peach. And there's kind of a spot, a groove in the middle where the messenger RNA can kind of nestle. Right, and this is where that shift from the sequence of bases is going to be made to the sequence of amino acids here at the ribosome. In order to make this switch from the base sequence language of nucleic acids to the amino acid sequence language of polypeptides or proteins, I have two problems. Well, not me, but my cells have two problems to overcome. First is a language issue, right? There aren't enough letters in the language of nucleic acids to have a one-to-one relationship. One base goes to one amino acid, right? There's only 4. And I got 20 amino acids that I need to code for. So I can't use 2. There's only 16 combinations of 2 of those 4 bases. And so when I move to 3, there are 64 different combinations possible, and that's more than enough, which is a good thing. We'll talk about that later on. So these 3 groups of 3 bases that correspond to a particular amino acid are called codons because they code for a particular amino acid. So your messenger RNA is actually read in groups of 3. Now, my other problem is I need some sort of translator, something that has both the language of amino acids and the language of nucleic acids in it. And I remember when I was in German class, I'd bust out my Langenscheid's dictionary, and I'd have the English words on one side, German words on the other side. And our cells have their own version of this. They have what are called transfer RNAs. And on one side of the transfer RNA is the base sequence. This is the complementary bases to the codon. We call this the anticodon. And on the other side of it is the amino acid itself that corresponds to the anticodon. So these transfer RNAs then are like the decoder rings that will actually make that switch between the languages. There is actually more RNA that we can talk about. Ribosomes themselves are made out of ribosomal RNA, and there's even more. Oh my Lord, so much RNA. Come see me in AP Biology. We'll talk about them. So we've talked about transcription, making messenger RNA just like through, it looks just like replication. It's like one-sided replication. And now we're getting to this next stage of making proteins, which is translation, actually making the conversion from the base pair sequence codons now into the amino acid sequence of the proteins. Again, we call this translation because now we're shifting languages. And it's important for us to associate translation with the ribosome, where we associate transcription with the nucleus. The next part of this process is called elongation, and it has to do with growing the polypeptide chain. If you notice down here at the ribosome itself, I have enough space for 1 and 2 codons to fit. That means that 2 transfer RNAs, here's one and here's the other, can bring their amino acids into position. Once those amino acids are in position, a covalent bond, a peptide bond more specifically, forms between the 2 amino acids. In doing so, what that does is it actually breaks the bond here. It breaks the bond here, all right, and frees that amino acid from its transfer RNA, and this whole messenger RNA carriages up shifts over one codon. And so this next codon back here, these guys here, which are coming up, will move up into this position, and this codon will move into this position. And the whole thing shifts over one. So this means in the past, this amino acid is associated with that codon. This amino acid used to go to that codon, this one to this codon, this one to this codon, and this one to this codon. All right, but it has carriaged all the way through. And this way, as that messenger RNA gets kind of yanked through the ribosome, another transfer RNA will come in and bring its amino acid into position. And so you get this kind of growing chain. That's why it's called elongation. The last step of the process is called termination, and that's when the polypeptide chain is done and it kind of breaks off and floats off and folds into its particular position, and the messenger RNA is free to be broken down or go to another ribosome and make a whole nother protein. For our purposes, we're going to associate, though a chain of amino acids is called a polypeptide most appropriately, for our purposes right now, we can really equate a polypeptide with a protein. They're not the same thing. There is a difference, but for our purposes right now, that's totally going to work. And for right now, I think that's enough. Thanks again for checking in to Mr. Ulrich's Land of Biology. If you have any questions, please drop them in Google Classroom, put them in the comments below, send me an email. You know how to get in touch with me. Other than that, we'll see you in class.